Good evening, ladies and gentlemen. This is the Mineral Water Group. Full cleaner of water with healing properties can be found all over the world dating back centuries. It has actually been so persistent through society that it is still used and even prescribed today in Korean society. Bathing in mineral springs to treat diseases is called polynosotherapy. One of the most famous mineral spring is the Blue Lagoon in Iceland, shown here. We hope to investigate whether or not uh, Ballinotherapy could be considered as major treatment of various diseases. We decided to narrow down the field of application for our project to burn victims. We could focus more on skin. We imaged a final product for mineral spring component as sort of typical cream that would be applied to the surface of the skin in case of minor burns. Because of this, we wanted to limit the cell types that we were using during the experimentation that have a major role in wound healing of skin. With construction, we decided to stick with fibroblast, which proved to be easy to work with, and keraticitis, which would be difficult to use, but it would be better representation of wound healing. The goal of our project was to develop a methodology to determine the in vitro biocompatibility and wound healing efficiency of mineral water-based media to treat damaged epithelial tissue. Our project had two main goals: characterization and methodology. We first wanted to characterize the physiochemical properties of the mineral water through using the environmental scanning electron microscope, energy dispersive X-ray spectroscopy, infrared spectroscopy, and go on. While knowing the physiochemical properties is important, we also had to actually design the methodology that would determine the wound healing capability of the mineral water.
Our characterization results showed us that the mineral water components consisted of a homogeneous silicon dioxide crystal in a cristobalite structure. A cristobalite structure is formed at high pressure and high temperatures.
Fibroblast cells were seated in a 6-well plate at a concentration of 50,000 cells per well. The experimental group was given media mixed with minerals from the water at a concentration of 0.01% weight. Once the monolayer was grown on the surface of the plate, a micropipette tip was used to create a small scratch on the surface. Each well contained 2 scratches. The cells were imaged immediately after the scratch was made and again after 24 hours. The cell migration rate was calculated using the equation shown on the left. The migration rates of the two groups were found to not be significantly different from one another. While this was a bit disappointing at first, since the mineral water media showed no effect on cell migration, we were able to conclude that the minerals are not toxic to the fibroblast cells since they do not hinder cell migration rates. The PDMS strips allowed us to create more control craters within the cell monolayers with equal lengths. We once again calculated the cell migration rate for the 3 intervals of 4 hours using the same equation. Once again, no significant differences were found between the two.
Our studies have established that the principal detectable component of the mineral water is homogeneous crystals of silicon dioxide in the cristobalite structure. We have shown that at a concentration of 0.01% by weight, the mineral water media is not toxic to fibroblast cells. Our pilot scratch study had some shortcomings such as consistency and imaging time that we were able to mend with the barrier assay. The barrier assay allowed us to control the size of the cell destructions and ensure that they remained uniform throughout the assay. Keratinocytes would be a more accurate representation of the skin cells that are involved with wound healing processes for burn wounds. We do recommend a few more experiments that are a bit more sophisticated than what we have already completed. The Transwell membrane assay is the most sophisticated assay that we could have realistically used for experimentation. This assay is commonly used to test the migratory effects of various agents on cells. Cells are allowed to form a monolayer and are then inverted over a semi-permeable membrane which contains the substance being tested, in our case the mineral water. Cells would then migrate through that membrane. We planned on electrospinning the membrane component of this assay and then incorporated the solid component of the mineral water. This assay would allow for a more accurate representation of what would occur if our mineral water component would be applied as a topical cream.
Moving away from cell migration rates, we investigated other cellular activities involved in wound healing and found a study regarding cell membrane impedance. Increased electrical activity in the cell membrane has been seen as a key factor in determining the presence of certain biological events, including division and growth in cells. Performing an experiment with a setup similar to the one shown below with our mineral media sample would allow us to determine if exposing the cells to the minerals affect their electrical potentials and therefore certain physiological processes.